without anything else, I want to introduce Vaughn Cooper, who very kindly agreed to be our leader today. And I know he's also facing some of the same challenges that I am with kids at the house and being cooped up and everything else. He's in Pittsburgh, not really in Hawaii, like it appears on his background that you'll see shortly. He's an evolutionary biologist and a microbiologist at the University of Pittsburgh in the School of Medicine. He's also the founding director of the Pittsburgh Center for Evolutionary Biology and Medicine, and we'd like to work with you, Vaughn, going forward as one of the organizations supporting this, and I think that'd be really great. We've asked our leaders to limit their presentations. I don't know exactly what we asked you, Vaughn, but something like 10 or 15 minutes, 5 minutes, whatever you want to do. And after that, we really encourage your participation and certainly start entering questions into the chat box along the way. And we'll wrap this up about 12:50. So we've got a good 40 minutes or so, for discussion. So thanks very much for joining us, Vaughn. Great to have you here.
Yeah, it's awesome. I was just, you guys can hear me? Is that right? Yep, okay. This is an incredible opportunity. We were chatting before we went live about how this crisis has actually been remarkable at lowering the barrier to entry into this community and the synergy among centers. And I think there are sort of two reasons I was really excited to join you guys today. The first was to kind of share my perspective about how this is a call to arms for our community, evolutionary medicine, because I think that we're doing a lot of great stuff, but we need some really big visible wins. And I can't imagine, frankly, a better opportunity for us to sort of make those wins visible, to achieve those victories and achieve them in a public manner for research, for education, and for teaching and outreach. And the second is just to share a few strategies, how we in the center, and I in particular, are trying to chip away towards those victories. So I put together a bunch of slides, which I'm going to share now. I won't share all of them. I'd like to be responsive to your questions or comments. So let me share screen first. All right, and let me see if I can see you guys. All right, I can see, can you see my screen, which is slides right now? Yes, all right, great. So yeah, this is the title that I sent to Meredith, before I get into a little bit of material, I want to introduce the center at Pittsburgh. It's been together since 2017. We kind of had a public release and a public ceremony in 2018. We have a website that you can see down below. There were actually 6 co-founders, and unfortunately one of our founding faculty left Pittsburgh. So our executive committee is 5, and we are Ann Carvunis, Dennis Koska, Eric Wright, Matt Nikotra, and myself. But we're also have 23 other faculty members from the University of Pittsburgh, not just in the medical school, but also many other departments, Carnegie Mellon University and Duquesne University. So all the sort of the 3 major research institutions in the city of Pittsburgh. It's a vibrant community. I guess one thing that I think sets us apart is that we are centered and supported by the medical school. So we're at Evolutionary Biology and Medicine that lives in medicine. And so we try and keep that focus at the fore about our research and our outreach. Just a couple things about what we're doing. So our mission, like many of you, is to advance the fields of both evolutionary biology and medicine by catalyzing research at their interface, and also to promote education opportunities at that interface. And so some of our actions are to sponsor seminars and an annual retreat. We've been teaching a course, our first course in evolutionary medicine, the 5 of us, which, like many of your courses, got disrupted. And so we've moved to online, and it's gone well. And that's the first course in a 2-course series that's an evolutionary medicine certificate for PhD programs that are hosted by the School of Medicine in Pitt. We have a Catalyst program where we're actually reviewing those grants right now, where we support pilot research that's proposed by a postdoc or a PhD student or an MSTP student working at the interface of evolution and medicine. And then we support outreach. And one of the outreach activities that, that is affiliated is one that I had founded 7-8 years ago called Evolving STEM that I'll mention towards the end. And so, okay, to the main part of my presentation. Again, I think this is a call to arms in this time, and what we all, I'm sure, are thinking is how can we contribute? We can contribute meaningfully by doing new research about viral evolution, about responses to viral evolution, about why we vary in our susceptibility to the disease and how we might vary in our recovery, about epidemiology, about sort of the notion of evolution epidemiology and how those two processes feed back upon one another, and then about education and outreach. And I'll focus a little bit on those. I just want to throw 2 other questions that maybe to provoke discussion. What can the ongoing evolution of SARS-CoV-2 teach us about the biology of the virus itself? And then what can this virus teach us more about generally about coronavirus evolution or about other viruses, viruses of human, viruses of livestock, and so on. So we're just starting to think about that, at least we, our group. So I want to just give a quick outline, some ideas, right? How can we contribute? Well, we all are presumably more trained in phylogenetics than the average researcher. And a lot of our understanding about the spread of this outbreak is rooted in the concept of phylogenetics and the theories of phylogenetics. And frankly, the public does not understand how that works. But by developing research and communication tools that explain this for a general audience, including the students that we're schooling at home, we're going to be building a framework that increases the dialogue and the knowledge of what's ongoing. We can also study the molecular evolution of the virus to understand maybe some forces of selection on the virus. And more recently, we've been starting, I think a lot of us are wondering why immunity differs with age. And then we as a field have been thinking about why immunity varies with age and other factors. You know, I mean, I think a good chunk of I see Randy's dialed in, a good chunk of Randy's original book with George Williams talked about that. We can talk about the evolution of virulence and the evolution of transmissibility. I just got a paper to review from a great group I suspect you'll see soon on theory about how transmissibility might be subject to selection during this pandemic. And so I want to just say, maybe take some notes right now and put some questions in the chat. How can we contribute in terms of research? I'll show you one bit of research that we've done. So this is work by Eric Wright and Seema Lakdwala, who's a flu virologist here in my department, and myself. And so what Eric did was download all of the genomes available as of a week ago and constructed a multiple alignment, built a tree, and then identified mutations across the SARS-CoV-2 genome. And so over here, if you can maybe see my cursor on the left, this is the numbers of independent substitutions. So these are mutations that we can reasonably infer are independent and not just spread by epidemic growth. And these are the different genes. And what we've highlighted are some regions in peach that are really underrepresented in the database, but also we've identified in these blue sections some particular sites that have mutated multiple times. And so we're getting ready to submit this paper soon. You know, this sort of research is available to any of us, and the data itself is evolving on the fly. And so our hope is that this analysis will show how different SARS-CoV-2 regions are subject to both purifying selection, the regions in the peach, and positive selection in blue. So there's kind of hardcore research that's come out really quickly from this opportunity. So maybe I'll pause and take any questions on this. And then I'll probably move on to the videos that we've done in the outreach. Let me see what's in the chat box here.
If anyone would like to ask a question while he's
Yeah,
looking at the chat box, you can use the raise hand function to do that.
Go ahead. Oh, you know what? I've lost the box. What happened? Oh, there it is. Okay. I don't see any questions in the chat. All right. I guess I'll maybe move on.
Looks like we did just get one question in the chat box from Lily Howell. Trevor Bedford once mentioned 12 mutations. How is their data compared to yours?
Yeah, so these data are, and I'll just skip ahead quickly on the slides. These data are the data that underlie Trevor Bedford and his colleagues' amazing site, nextstrain.org. And I used the data actually that came from here. Next slide. This is from the GISAID website, which has, as of this morning, 7,108 full genomes. And this is a giant phylogeny showing these different clades. In any case, the question about 12 mutations are on. There are some over here on the right, there are some lineages that have maybe as many as 20 different mutations. There are some lineages that are maybe only 1 or 2 mutations different from the original strain that was sequenced from Wuhan. And then what you can see on average, the average genome will have on the order of about 8 mutations as of today. But there's quite a bit of spread. And you can look at that differently over here on the left by looking at how these different clades have emerged basically by geographic spread. So I think the major take-home message that we need to be sharing is that most of the mutational diversity is informative for where the viruses came from by geographic spread, but not functional differences. It's possible, we hypothesize that some of those mutations that I showed in the last slide may be functional, but they're probably, we certainly have a lot more to learn, I guess. All right.
Looks like we do have another question from Joseph Graves in the chat box as well. With those numbers, are you separating ensign? I'm not sure if I'm pronouncing that correctly. Ensign and sign mutations. So
non-synonymous and synonymous mutations.
Okay, thank you.
Yeah, so I just went back to the original the new figure that we've put together, and the mutations are colored differently, and the ones in blue are non-synonymous. The ones in peach are non-coding, or sort of pink are non-coding. The synonymous mutations are in brown, and they are maybe a little harder for you to see. There's a few sites where you see both synonymous and non-synonymous changes. So like that U13402G, that site is both synonymous and non-synonymous because that site is read 2 different ways. So it's overlapping reading frame. I hope that answers the question. So the mutations we're highlighting there in the middle are actually non-synonymous, repeated non-synonymous substitutions that affect the protein sequence of the virus. All right. If it's okay, I'm gonna skip ahead to some of the educational opportunities or outreach opportunities. As I want to say, this is preliminary, certainly not even yet a preprint, but I wanted to share that, you know, what's amazing about this time is the data and the tools allow us to do really rapid studies of evolutionary change on the fly. And I want to again acknowledge, you know, this field is not new. That review that I cite down in the lower left from Moya et al. Is from 2004. It's one of the probably best cited reviews that uses the word evolutionary epidemiology in the title, but that field is much older than that. It goes back, you know, certainly back to when Paul Ewald was talking about it in 94, and probably much before that, probably into the 80s, and maybe before that. There's probably people on here who can correct my history. Okay, so how can we contribute in education? Well, as you all know, we're probably in the thick of it. Remote learning is placing huge demands on teachers like us for relevant, engaging content, and everybody wants to learn about COVID. So for better or worse, this is a great opportunity to learn some pretty useful lessons in population biology and phylogenetics and virology and so on. And I'd love to hear other people's ideas about this. And one reason I'm really keen to hear about your ideas is that the American Society of Microbiology is curating some of these resources and will be posting them probably this week on their main website to provide kind of a shared resource for vetted information and recommended material for teachers. And so my recommendation is that as you're putting your material together, if you're not already, post your slides on Figshare, and maybe we can use a tag that aggregates our materials in evolutionary medicine. That would be a great opportunity for our centers to coordinate, and then also share your video recordings on YouTube or Vimeo. And then share them on social media at your own risk. I think you'll find that you'll mostly be engaging in productive discussion with family, friends, and the public. The big motivation for me to put these couple of YouTube videos out was I was getting a bunch of questions from various folks, colleagues and others, and family about mutational diversity and what it meant. And I felt, well, this is something that I actually can say something about, so I will. And so you probably saw links. I put together a couple of YouTube videos. And the first one I talked about really what is phylogenetics. And if we've learned anything about YouTube, cat videos sell. And so I put cat phylogeny in the video to start. And on the down below, you'll see here that I just have a nice family tree of cats. And it shows kind of one point that I found was pretty cool, that snow leopards are actually more closely related to tigers than they are to leopards. And that this clade of cats diverged around 12 million years ago. Well, we can apply the same logic to the coronaviruses. And over here is just the text that I put in to promote the YouTube video. And then I provided a link to the slides, which a bunch of teachers have gone ahead and downloaded and used in their lectures. And so you probably know that SARS-CoV-2 is related to SARS-CoV, the original SARS virus. All of these in red are human coronaviruses. This is the MERS coronavirus, and now I'm just going to animate in the likely animal sources of these viruses. And basically, you'll see that bats are the common ultimate sources of these viruses, even if there was an intermediate host that is not yet resolved. And you all know about that the civet was the immediate animal host for the original SARS virus. We still, I don't think, have heard a clear sense of what the original, the approximate animal host was. So despite some claims about the pangolin, that's still not yet resolved as far as I know. And then the second video, I wanted to talk about, really address a bunch of comments that I'd seen in social media about all these strains being, making it impossible to design effective vaccines. And what I pointed out is that, you know, there's lots of mutations, and mutations happen in RNA viruses, but maybe the thing that we can learn the most about is where mutations aren't. And so I use this lesson that came from social science, up here on the left, the schematic of where bullet damage was not found in a fleet of World War II bombers that safely returned, and the absence of those bullet holes is because you know, when a bomber was hit in those sites, the bomber didn't come home, right? And so those sites, I think, are where the bomber was most vulnerable, and consequently we can use the same analysis. And this is some new analysis I did quick and dirty for the video, showing that in the polymerase regions, mutations are underrepresented. And that's a good thing, because the polymerases are drug targets for a bunch of antivirals that are part of trials. I really like this analogy. I think it's maybe the best analogy for teaching the concept of purifying selection or negative selection. So I'd encourage you to maybe use that, or come up with your own and share your own analogies about how we can teach tricky concepts in evolutionary biology. And so just last point before I stop and take some more, open up for dialogue. We've been running this evolution and action curriculum in almost 20 high schools for the last several years. And the shutdown happened just as a bunch of our schools were scaling up to do the experiments concurrent with the Revolution curriculum in their high school biology classes. And we've been cooking up a bioinformatics module to accompany that, because we can sequence the mutants that the students evolve. So we are actually going to be releasing that curriculum content today to a bunch of our teachers, to have them download FASTQ sequences, have the students use an online tool called Patrick to call the variants and learn how we can use sequencing to identify the genetic basis of evolved changes in bacteria. So I kind of said a lot there. I have more slides where I can talk about it, but I'd prefer to stop blathering and open this up for discussion.
Yeah, so if anyone has questions, feel free to use the raise hand function. Looks like we have one from Jay, or hand went back down. Jay, did you have a question?
Yeah, I'm sorry, I didn't know whether it would actually had raised.
Yeah.
Thanks very much for this. And particularly, I looked at your two videos before this, and I thought the bomber model that you were using was really effective. And I think that was very good. A couple of things that as I was thinking about this, for those of you who don't know, I'm the chair of ISEM's Committee on Education and Outreach. One of the things that I thought you might want to think about changing, this is just sort of a general comment that might be useful for other people, on where you were starting to get into math. You said math alert, and what I worry about is that when you say math alert, that just reinforces the idea that math is really scary and really hard. So I would either suggest taking that out, or put in and change math alert to math opportunity. Okay, because I think that, you know, students have to understand that math is an integral part of what it is that we're doing here, and that math actually has some meaning. And I think the way that math is taught too often, they see no connections. So the other thing I would suggest then, when I was listening to that discussion, which I thought was quite good, I also thought that depending on the audience, it may be a little bit too far over their heads. So it might be useful, since you have a lot of resources at the University of Pittsburgh, and I would say this would be true for almost anybody, to try to think about how to interact with people in other departments who have that kind of expertise. So people in the math department, or possibly in biomathematics. There's a guy at University of Pittsburgh by the name of Sam Donovan in biology.
Yeah, Sam's great.
Yeah, who might be able to do something like that. And I think maybe provide some supplemental materials, because again, when people are doing this online, it's really hard to gauge where students are. And there may be some students who understand log 2, you know, and that sort of thing. But for a lot of other students, it's just going to kind of sail past them and reinforce the point that math is just too difficult to understand. So there might be some way to be able to get some other people to help to think about how they could begin to do this more effectively. There's also a group funded by the National Science Foundation called NIMBUS. It comes out to NIMBIOS, but they pronounce it NIMBUS, the National Mathematical Biological Center. And I think that they would have some good information there too about how to help with that. But math is a real issue, and I just worry about that. Thanks.
Yeah, thanks, Jay. That's you said a lot, and I mean, I think the one point that I want to echo is there's value in putting out a free to reuse, recycle, revise first draft for communications, like for ideas like this. And you're absolutely right. Boy, it's impossible to hit all audiences with one video. And your point about the math alert, second point I wanted to just touch on. The feedback that I've gotten about that video has been polarizing, meaning that they've either really liked the fact that I went to the math or really disliked that I went to the math. So that shows that we either have to come up with two different strategies or multiple different strategies to tackle that, or to do it really differently. And even though I'm trained in population genetics, talking about that math is actually even hard for me to think about, let alone to discuss. So
I agree with you. It's really difficult. And when I was a professor, when I used to ask my students, which was at a liberal arts undergraduate college, why they decided to become bio majors, a lot of them said because didn't have to do math. You know, if I wanted to do math, I'd be a chemistry or physics major. So it just seems that what we need to be thinking about is that students have been really socialized into thinking that math is irrelevant, really difficult, and for a lot of reasons they're right, given the way that it's taught in too many places. So I think we just have to be much more aware of the various audiences who may be doing, particularly on something like evolution, where just the word raises people's hackles.
Yeah.
You know, which is why Charlie was talking about the Zoom bombing. I mean, there are people who are probably likely to do something like that who are listening into this, who are not particularly amenable to what it is we're talking about. Thanks.
Thank you. Yeah, I would love to see how to do this better, and I'd love everybody's input about revising and producing new versions of this, particularly as the data themselves change.
So going back to the chat box, it looks like there's a lot of questions that revolve kind of around the genetic diversity of SARS-CoV-2.
Yep.
There was a question a while back about assuming we develop a vaccine for this virus, do we expect it to be an annual vaccine like we see with the flu? And if not, why is SARS-CoV-2 different?
Yeah. Okay, so yeah, well, I don't know if I'm still sharing, right? So you can see the slide. Okay, so that's an awesome question, and the short answer is I don't know. The long answer is that I'm working on another video that's going to be a lot harder than these first two about that. The data are okay. So here's what I can see from experts I trust. I mean, like proper immunologists. We do generally get immunity to coronaviruses that lasts a year or two, maybe more. We don't know how much of that loss of immunity is due to loss of training of our sort of deadening of our immune response, sort of resensitization of our immune response versus changes in the virus. My hunch is that it's more immunity than the virus. We actually don't know that much about what factors in the virus promote immunity best. So that's a big question. So the root of that question was, how does evolving variation in the virus play against susceptibility and immunity? And we don't know, because we don't know what factors of the virus contribute to lasting immunity. I think there's good evidence that the spike is a big part of it. We see one mutation that's in spike, actually maybe two. One of them is non-synonymous. One is synonymous, but they're pretty rare relative to some of the others. That's what we know so far. Working on it.
Great. There's also a lot of discussion in the chat box about virulence and transmissibility trade-offs.
Yeah.
From Michael Reiskin, he's wondering, are there any of the sequences being linked with clinical information? So do we have any information on specific genetic variants of this virus that are making it more virulent?
So I've had a couple of people send me links to preprints on medRxiv or bioRxiv talking about that. So far, I haven't seen anything that's looks like it's going to hold up, to be frank. But it's right. I think still at this stage, it's super hard to decouple epidemic spread from, which is really driven by us, right, by human population biology, than from genetic variation. I think we're going to learn more as time plays out and as distancing holds in some places. When distancing is released, again, it becomes more about epidemiology than it does genetic diversity. The paper that I am, one of these things that I'm reviewing points out that, conditions are favorable for traits that affect transmission to evolve. For example, a mutation that increases the asymptomatic period would be favored under most conditions because it should promote transmission. So theory says that conditions are right. We just don't know if the genetic variation is such that it could happen. And I also want to say that in my first video, when I said that most of the variation doesn't matter very much for function, I meant it because I didn't think the population size of the virus was high enough for selection to be actually acting on heritable variation. That's certainly changed in the last few weeks.
Hey, Vaughn, I have a question related to One Health. And in one of your first slides, you had mentioned how can we contribute in the field. And one of the ideas I had was One Health, but I was just wondering what you think and what other people think about the relationship between One Health and evolutionary medicine. And sort of as a related point, what are some of the ways that we can contribute to the One Health perspectives that are out there, bringing in a firmer evolutionary or ecological perspective? So it's sort of two questions, how do they relate right now, and what are some of the ways that we can really contribute to strengthening, understanding within the One Health lens?
So awesome question, Charlie. And I think there's probably people connected here who are better to address that question than I am. Their coronaviruses have many hosts, including some in affecting valuable livestock. And I'm wondering, boy, I wish I had a colleague at a vet school to talk about right now. Maybe there's somebody on right now who has some perspective. But in general, I think folks focused on pathogens from a One Health perspective are frankly going to have better perspective on viral adaptation to new hosts than we do right now. We in human medicine.
Mhm.
So looking in the chat box now, kind of from that One Health perspective, I'm seeing that part of the clinical description in China included diarrhea, but we haven't heard much about this recently. And last week we had a discussion on One Health and SARS-CoV-2. The question is, could ancestral strains have shifted away from a fecal-oral transmission if these coronaviruses are of an enteric nature in bats?
Yeah, so I see Michael posted that. That's a good one. Yeah, there's been varied discussion about the GI role. I think it's clear that you can get viable virus from stool up to 14 days post basically past the end of fever. Which is pretty alarming. So I think the fecal-oral route remains, but I don't know about you're right about there's been less discussion about the symptomology of it.
All right, it looks like also in the chat box, there's a mention that there's an old TB vaccine that confers some benefits during this pandemic. I wasn't familiar with that. Do you have any thoughts on how that might be happening?
So I should know this. So the argument is that BCG, people who have had the BCG vaccine against TB are going to have some protective immunity. And I think there are some studies, certainly some preprints talking about that. I don't know the latest, I'm sorry. Is anyone else here?
Hey, Vaughn, I have another question related to your first video.
Yeah.
You mentioned in there bats, and if I recall, you had 3 arguments for why so many emerging diseases come from bats.
Oh yeah.
One is that 1/5 of mammals are bats, which is a good argument. Another, I think you had, and you might have to correct me if I'm wrong, but it had something to do with flight. Oh, it looks like you're opening your slide.
Yeah, I'm going to load it. I had it up, so I wanted to. Let me see.
And then a third was, I can test myself here. I think the third one had to do with tolerance, basically, right? Some kind of a tolerance mechanism.
Yep. So I just put the old slide for the video up here. Yeah, so why are bats the reservoir for coronaviruses? I spent a lot of time digging into literature about this, and it's an incomplete literature. But basically, there are a lot of species of 20% of mammalian species are bats. So there's one. Although a paper was just posted in PNAS yesterday, which I think partially debunks that explanation. And I haven't even read it yet. But it does point out that viral diversity associates with both species diversity, but also the population sizes of the host species. The other explanation is the flight argument. And because flight is costly, they've got, and it's not just that, they're actually super long-lived. So combination of selection for longevity and the ability to tolerate a lot of oxidative damage means that bats do a better job of dealing with sort of defending against byproducts of metabolism than they do, and that's been shown experimentally. And then the third point is there's been a couple of really cool articles showing that their antiviral responses through the sting-mediated pathway are dampened. So they have a partial loss of function mutation that's common in some species that dampen the innate response to viral infection. So that mutes that initial immune-related damage prompted by innate immunity. So those are the 3 things I came up with, and I put links to those articles at the end of that video.
That's great. I mean, it seems another possibility is that, at least for many bats, they live in large colonies. They have
Exactly.
Really large aggregations of animals, and that may allow, you know, for them to carry more infectious diseases, right? To, you know, just have more infectious agents. And so that's more of a kind of an exposure argument. And I often try to think, you know, when I'm teaching my students, I emphasize their exposure pathways. You first have to think about how does exposure work, and
Yep.
then also how does susceptibility work.
Yep.
So I wonder if people have ever, you know, mentioned that aspect of it in the case of bats.
Right. So it seems like there's, you know, some great opportunity for comparative biology among bats based on the size of their roosts and the size of their colonies during hibernation and so on, Charlie.
Yeah.
I wish I knew more.
We should have asked
Again,
Kevin Olival last week.
Was that?
We had Kevin Olival on last week. We should have asked
Yeah,
him.
I missed it. I'm sorry.
Thank you.
Yeah. I did see in the chats that there's some desire for me to show a bit more about the education program. So let me just quickly skip to that slide. Okay, so the program is called Evolving STEM. The website is evolvingstem.org. I'll send a link to Meredith to distribute with this video too. And if you're interested in sharing this bioinformatics curriculum we've put together based on this program, basically students, hopefully you can quickly see, students do an experiment in their high school classroom, and they collect mutants. So at the outset, they have very little variation over on the left. And after 5 days of selection, the population diversifies. They pick a mutant, we sequence the genome, we identify the mutation, and so the bioinformatics module starts after that. And so please just go to evolvingstem.org there and fill out the contact us form, and we can send you a link to all of those materials. We are shipping it in alpha, just like my YouTube videos. They are meant for discussion and revision. So we really would like your input about how it works. And we don't have you know, a rigorous set of assessments for them yet. We rely on you guys as teachers to do with it what you will and share what you've learned from it.
Okay, Meredith, any other questions that are coming up? Or anyone else have a question? You can raise your hand or type it into the box. Any other questions or comments?
Okay, so I see one here most recently about how does interferon work? Yeah. Got someone who wanna jump in on that? And some discussion about diarrhea as well.
Well, I'll jump in on the discussion of diarrhea because that's relatively simple. And the point I made is that there are population genetic differences between Chinese, Europeans, and sub-Saharan Africans. In particular, as I'm sure everybody knows, sub-Saharan Africans have much higher HLA diversity than other populations in the world. In addition, we know the microbiomes of these populations differ as well. So it'd be extremely difficult, at least with the current data, to be able to tell whether the absence of diarrhea as a symptom is the result of the virus's evolution or a result of who the virus is infecting. So that's the point I made.
Yeah, thank you.
Okay, any last quick question? I think we're about at the end of the hour here, but if anybody has a last quick question, we can take it.
Hey, Kevin, I see your hand up.
Oh, good. Here. Yeah, you know, again, thanks. What a great job you're doing. Can you discuss the phylogenetic relationship to this pandemic and the Spanish flu in terms of, you know, phylogeny to the avian virus that caused the Spanish flu? And also, the Spanish flu did not originate in Spain, right? I mean, I've heard that it started in Kansas because of World War II World War I recruits being crammed into barracks. Do you know anything about that? It, you know, it really did originate in China, right? This one.
Okay, so I'm sorry, my internet just paused out, so I didn't get everything that you asked, but I think you were talking about the phylogeny of these viruses versus what, and then you also brought up the 1918 influenza, right? So concerning,
Yes.
is that right? Yeah, so
Yeah.
concerning this virus, it definitely was isolated in Wuhan, China, and all of the subsequent, it looks like there was a single point of crossover, single patient, and then followed that, there's been continuous person-to-person spread. So the phylogeny, I'll go back to Trevor, the next strain. So what this is showing in two different ways is that all viruses trace back to a single source, and that single source genome is, Wuhan 1, basically. Concerning, and then that strain is very closely related to some strains that were isolated from pangolins and then also from horseshoe bats. But the immediate ancestor, whether there was a strain in another species in between, is still not known. So we can't pin it
What
on
about
pangolins.
a most recent common ancestor? Between the avian virus that caused the Spanish flu and the Wuhan virus that, what is the phylogenetic relationship between those two viruses? One came from a bat and the other one came from a bird, right?
Yeah, so I mean, the influenza viruses and coronaviruses are as different from one another as any two different visible organisms you can imagine. They're incredibly different viruses, so they're entirely unrelated.
Is everybody sure that H1N1 1918 was actually from birds? Because I thought it was from swine.
I think the model is that it was both birds and swine, right? Like most flu viruses.
Yeah, that was my understanding.
Yeah.
Yeah, not
Yep.
just from birds.
And did it really originate in Kansas in the barracks of new recruits to World War I?
What's really clear is
that?
that the 1918 pandemic definitely started in the United States and swept through our own barracks first, and then into the civilian population, and then into American soldiers who were stationed in Europe. And take-home lesson of that pandemic is that one of the reasons why it was so devastating, besides the fact that we don't have the science and medicine that we have today, was that the political leadership of the United States, particularly Woodrow Wilson, lied to the American people. And the Public Health Service continued lying to the American people. And so no one was able to take precautions to protect their lives. John's book was a really excellent read concerning that.
Well, I'm just curious. I have one more question, if you wouldn't mind.
Well,
it's just
probably
like
close up, Kevin. I'm sorry.
Okay.
Okay. But, if you want to contact Vaughn directly or throw it out in the chat while I'm wrapping up, that's totally fine. Just to sort of have it on the record so everybody can see it. That'd be fine. But we do have to wrap up, and I just wanted to thank Vaughn for coming. You didn't really go anywhere, I guess, except your office. But thank you for joining us. Thank you so much for being a part of this discussion, and also for your videos, which I know are circulating. And I hope you're going to keep us posted if you have any others come out. For example, on the immune system, I think you're talking about that.
Yeah, thank you. I'm really honored again by the opportunity to see so many of you and talk with many of you. I hope that you'll follow up if you have any ideas or your own resources. I'm really excited to to continue just collating resources. I'm sure I can learn from you, and hopefully I can help you share your own outreach and teaching materials and research materials, for that matter, with the broader community. Again, I think this is an incredible opportunity for our community to make a difference.
Excellent. And thank you, Meredith, for organizing us and keeping things running so nicely. And thank all of you for joining us. This has really been fantastic. And stay tuned for the next one of these, hopefully next week or the week after. If you're not already on our list, you can go to the TRICEM website. That's TRICEM, tricem.org, and you can sign up. So thank you all for joining. Thanks very much.